A real-time polymerase chain reaction is a laboratory technique of molecular biology based on the polymerase chain reaction, which is used to amplify and simultaneously detect or quantify a targeted DNA molecule. The procedure follows the general principle of polymerase chain reaction. Its key feature is that the amplified DNA is detected as the reaction progresses in real time. This is a new approach compared to standard PCR, where the product of the reaction is detected at its end. Two common methods for the detection of products in quantitative PCR are non-specific fluorescent dyes that intercalate with any double-stranded DNA, and sequence-specific DNA probes consisting of oligonucleotides that are labeled with a fluorescent reporter, which permits detection only after hybridization of the probe with its complementary sequence to quantify messenger RNA and non-coding RNA in cells or tissues. The MIQE guidelines propose that the abbreviation Qpcr be used for quantitative real-time PCR and that a tQpcr be used for reverse transcription Qpcr. 1. The acronym RT-PCR commonly denotes reverse transcription polymerase chain reaction and not real-time PCR, but not all authors adhere to this convention. Background. Cells in all organisms regulate gene expression by turnover of gene transcripts. The amount of an expressed gene in a cell can be measured by the number of copies of an mRNA transcript of that gene present in a sample. In order to robustly detect and quantify gene expression from small amounts of RNA, amplification of the gene transcript is necessary. The polymerase chain reaction is a common method for amplifying DNA. For mRNA-based PCR, the RNA sample is first reverse transcribed to cDNA with reverse transcriptase. In order to amplify small amounts of DNA, the same methodology is used as in conventional PCR using a DNA template, at least one pair of specific primers, deoxyribonucleotides, a suitable buffer solution and a thermostable DNA polymerase. A substance marked with a fluorophore is added to this mixture in a thermal cycler that contains sensors for measuring the fluorescence of the fluorophore after it has been excited at the required wavelength, allowing the generation rate to be measured for one or more specific products. This allows the rate of generation of the amplified product to be measured at each PCR cycle. The data thus generated can be analyzed by computer software to calculate relative gene expression in several samples. Quantitative PCR can also be applied to the detection and quantification of DNA in samples to determine the presence and abundance of a particular DNA sequence in these samples. This measurement is made after each amplification cycle, and this is the reason why this method is called real-time PCR. In the case of Rna quantitation, the template is complementary DNA, which is obtained by reverse transcription of ribonucleic acid. In this instance, the technique used is quantitative RT-PCR or qRT-PCR. Quantitative PCR and DNA microarray are modern methodologies for studying gene expression. Older methods were used to measure mRNA abundance, differential display, RNA-seq protection assay and northern blot. Northern blotting is often used to estimate the expression level of a gene by visualizing the abundance of its mRNA transcript in a sample. In this method, purified RNA is separated by agarose gel electrophoresis, transferred to a solid matrix, and probed with a specific DNA or RNA probe that is complementary to the gene of interest. Although this technique is still used to assess gene expression, it requires relatively large amounts of RNA and provides only qualitative or semi-quantitative information of mRNA levels. Estimation errors arising from variations in the quantification method can be the result of DNA integrity, enzyme efficiency and many other factors. For this reason, a number of standardization systems have been developed. Some have been developed for quantifying total gene expression, but the most common are aimed at quantifying the specific gene being studied in relation to another gene called a normalizing gene, which is selected for its almost constant rate of expression. These genes are also called housekeeping genes as they are usually involved in the functions related to basic cellular survival, which normally implies constitutive gene expression. This enables researchers to report a ratio for the expression of the genes of interest divided by the expression of the selected normalizer, thereby allowing comparison of the former without actually knowing its absolute level of expression. The most commonly used normalizing genes are those that code for the following molecules: tubulin, glyceraldehyde 3-phosphate dehydrogenase, albumin, cyclophilin, and ribosomal RNAs. Basic principles. Quantitative PCR is carried out in a thermal cycler with the capacity to illuminate each sample with a beam of light of a specified wavelength and detect the fluorescence emitted by the excited fluorophore. The thermal cycler is also able to rapidly heat and chill samples, thereby taking advantage of the physicochemical properties of the nucleic acids and DNA polymerase. The PCR process generally consists of a series of temperature changes that are repeated 25–40 times. These cycles normally consist of 3 stages. The first, at around 95 degrees Celsius, allows the separation of the nucleic acid (1 registered trademark S double chain. The second, at a temperature of around 50–60 degrees Celsius, allows the binding of the primers with the DNA template. The third, at between 68–72 degrees Celsius, facilitates the polymerization carried out by the DNA polymerase. Due to the small size of the fragments, the last step is usually omitted in this type of PCR as the enzyme is able to increase their number during the change between the alignment stage and the denaturing stage. In addition, some thermal cyclers add another short temperature phase lasting only a few seconds to each cycle, with a temperature of, for example, 80 degrees Celsius, in order to reduce the noise caused by the presence of primer dimers when a non-specific dye is used. The temperatures and the timings used for each cycle depend on a wide variety of parameters, such as the enzyme used to synthesize the DNA, the concentration of divalent ions and deoxyribonucleotides in the reaction, and the bonding temperature of the primers. Classification. The type of quantitative PCR technique used depends on the DNA sequence in the samples. The technique can either use non-specific fluorochromes or hybridization probes. Quantitative PCR with double-stranded DNA binding dyes as reporters. A DNA binding dye binds to all double-stranded DNA in PCR, causing fluorescence of the dye. An increase in DNA product during PCR therefore leads to an increase in fluorescence intensity and is measured at each cycle, thus allowing DNA concentrations to be quantified. However, dsDNA dyes such as SYBR green will bind to all dsDNA PCR products, including non-specific PCR products. This can potentially interfere with, or prevent, accurate quantification of the intended target sequence. The SYBR green is excited using blue light and it emits green light. The reaction is prepared as usual, with the addition of fluorescent dsDNA dye. The reaction is run in a quantitative PCR instrument, and after each cycle, the levels of fluorescence are measured with a detector. The dye only fluoresces when bound to the dsDNA. With reference to a standard dilution, the dsDNA concentration in the PCR can be determined. This method has the advantage of only needing a pair of primers to carry out the amplification, which keeps costs down. However, it is only possible to amplify a product using a chain reaction. Like other quantitative PCR methods, the values obtained do not have absolute units associated with them. As described above, a comparison of a measured DNA-RNA sample to a standard dilution will only give a fractional ratio of the sample relative to the standard, allowing only relative comparisons between different tissues or experimental conditions. To ensure accuracy in the quantification, it is usually necessary to normalize expression of a target gene to a stably expressed gene. This can correct possible differences in RNA quantity or quality across experimental samples. Fluorescent reporter probe method. Fluorescent reporter probes detect only the DNA containing the probe sequence. Therefore, use of the reporter probe significantly increases specificity, and enables quantification even in the presence of non-specific DNA amplification. Fluorescent probes can be used in multiplex assays for detection of several genes in the same reaction based on specific probes with different colored labels, provided that all targeted genes are amplified with similar efficiency. The specificity of fluorescent reporter probes also prevents interference of measurements caused by primer dimers, which are undesirable potential byproducts in PCR. However, fluorescent reporter probes do not prevent the inhibitory effect of the primer dimers, which may depress accumulation of the desired products in the reaction. The method relies on a DNA-based probe with a fluorescent reporter at one end and a quencher of fluorescence at the opposite end of the probe. The close proximity of the reporter to the quencher prevents detection of its fluorescence. Breakdown of the probe by the 5' to 3' exonuclease activity of the Taq polymerase breaks the reporter-quencher proximity and thus allows unquenched emission of fluorescence, which can be detected after excitation with a laser. An increase in the product targeted by the reporter probe at each PCR cycle therefore causes a proportional increase in fluorescence due to the breakdown of the probe and release of the reporter. The PCR is prepared as usual, and the reporter probe is added. As the reaction commences, during the annealing stage of the PCR both probe and primers anneal to the DNA target. Polymerization of a new DNA strand is initiated from the primers, and once the polymerase reaches the probe, its 5' 3 exonuclease degrades the probe, physically separating the fluorescent reporter from the quencher, resulting in an increase in fluorescence. Fluorescence is detected and measured in a real-time PCR machine, and its geometric increase corresponding to exponential increase of the product is used to determine the quantification cycle in each reaction. Fusion temperature analysis. Qpcr permits the identification of specific, amplified DNA fragments using analysis of their melting temperature. The method used is usually PCR with double-stranded DNA binding dyes as reporters and the dye used is usually SYBR green. The DNA melting temperature is specific to the amplified fragment. The results of this technique are obtained by comparing the dissociation curves of the analyzed DNA samples. Unlike conventional PCR, this method avoids the previous use of electrophoresis techniques to demonstrate the results of all the samples. This is because, despite being a kinetic technique, quantitative PCR is usually evaluated at a distinct endpoint. The technique therefore usually provides more rapid results and/or uses fewer reactants than electrophoresis. If subsequent electrophoresis is required, it is only necessary to test those samples that real-time PCR has shown to be doubtful and/or to ratify the results for samples that have tested positive for a specific determinant. Quantification of gene expression. Quantifying gene expression by traditional DNA detection methods is unreliable. Detection of mRNA on a northern blot or PCR products on a gel or southern blot does not allow precise quantification. For example, over the 20 to 40 cycles of a typical PCR, the amount of DNA product reaches a plateau that is not directly correlated with the amount of target DNA in the initial PCR. Quantitative PCR can be used to quantify nucleic acids by two common methods, relative quantification and absolute quantification. Absolute quantification gives the exact number of target DNA molecules by comparison with DNA standards using a calibration curve. It is therefore essential that the PCR of the sample and the standard have the same amplification efficiency. Relative quantification is based on internal reference genes to determine fold differences in expression of the target gene. The quantification is expressed as the change in expression levels of mRNA interpreted as complementary DNA. Relative quantification is easier to carry out as it does not require a calibration curve, as the amount of the studied gene is compared to the amount of a control housekeeping gene. As the units used to express the results of relative quantification are unimportant, the results can be compared across a number of different RT-qPCR. The reason for using one or more housekeeping genes is to correct non-specific variation, such as the differences in the quantity and quality of RNA used, which can affect the efficiency of reverse transcription and therefore that of the whole PCR process. However, the most crucial aspect of the process is that the reference gene must be stable. The selection of these reference genes was traditionally carried out in molecular biology using qualitative or semi-quantitative studies such as the visual examination of RNA gels, northern blot densitometry or semi-quantitative PCR. Now, in the genome era, it is possible to carry out a more detailed estimate for many organisms using DNA microarrays. However, Research has shown that amplification of the majority of reference genes used in quantifying the expression of mRNA varies according to experimental conditions. It is therefore necessary to carry out an initial statistically sound methodological study in order to select the most suitable reference gene. A number of statistical algorithms have been developed that can detect which gene or genes are most suitable for use under given conditions. Those like GeNorm or best Qpcr can compare pairs or geometric means for a matrix of different reference genes and tissues. Modeling. Unlike endpoint PCR, real-time PCR allows quantification of the desired product at any point in the amplification process by measuring fluorescence. A commonly employed method of DNA quantification by quantitative PCR relies on plotting fluorescence against the number of cycles on a logarithmic scale. A threshold for detection of DNA-based fluorescence is set slightly above background. The number of cycles at which the fluorescence exceeds the threshold is called the threshold cycle or, according to the MIQE guidelines, quantification cycle. During the exponential amplification phase, the quantity of the target DNA template doubles every cycle. For example, a DNA sample whose CQ precedes that of another sample by 3 cycles contain 23 equals 8 times more template. However, the efficiency of amplification is often variable among primers and templates. Therefore, the efficiency of a primer-template combination is assessed in a titration experiment with serial dilutions of DNA template to create a standard curve of the change in CQ with each dilution. The slope of the linear regression is then used to determine the efficiency of amplification, which is 100% if a dilution of 1:2 results in a CQ difference of 1. The cycle threshold method makes several assumptions of reaction mechanism and has a reliance on data from low signal-to-noise regions of the amplification profile that can introduce substantial variance during the data analysis. To quantify gene expression, the CQ for an RNA or DNA from the gene of interest is subtracted from the CQ of RNA DNA from a housekeeping gene in the same sample to normalize for variation in the amount and quality of RNA between different samples. This normalization procedure is commonly called the Ict method and permits comparison of expression of a gene of interest among different samples. However, for such comparison, expression of the normalizing reference gene needs to be very similar across all the samples. Choosing a reference gene fulfilling this criterion is therefore of high importance, and often challenging, because only very few genes show equal levels of expression across a range of different conditions or tissues. Although cycle threshold analysis is integrated with many commercial software systems, there are more accurate and reliable methods of analyzing amplification profile data that should be considered in cases where reproducibility is a concern. Mechanism-based Qpcr quantification methods have also been suggested, and have the advantage that they do not require a standard curve for quantification. Methods such as MAK2 have been shown to have equal or better quantitative performance to standard curve methods. These mechanism-based methods use knowledge about the polymerase amplification process to generate estimates of the original sample concentration. An extension of this approach includes an accurate model of the entire PCR reaction profile, which allows for the use of high signal-to-noise data and the ability to validate data quality prior to analysis. Applications. There are numerous applications for quantitative polymerase chain reaction in the laboratory. It is commonly used for both diagnostic and basic research. Uses of the technique in industry include the quantification of microbial load in foods or on vegetable matter, the detection of GMOs and the quantification and genotyping of human viral pathogens. Diagnostic uses. Diagnostic quantitative PCR is applied to rapidly detect nucleic acids that are diagnostic of, for example, infectious diseases, cancer and genetic abnormalities. The introduction of quantitative PCR assays to the clinical microbiology laboratory has significantly improved the diagnosis of infectious diseases, and is deployed as a tool to detect newly emerging diseases, such as new strains of flu, in diagnostic tests. Microbiological uses. Quantitative PCR is also used by microbiologists working in the fields of food safety, food spoilage and fermentation, and for the microbial risk assessment of water quality and in public health protection. The antibacterial assay virtual colony count utilizes a data quantification technique called quantitative growth kinetics that is mathematically identical to Qpcr, except bacterial cells, rather than copies of a PCR product, increase exponentially. The Qgk equivalent of the threshold cycle is referred to as the threshold time. Uses in research. In research settings, quantitative PCR is mainly used to provide quantitative measurements of gene transcription. The technology may be used in determining how the genetic expression of a particular gene changes over time, such as in the response of tissue and cell cultures to an administration of a pharmacological agent, progression of cell differentiation, or in response to changes in environmental conditions. It is also used for the determination of zygosity of transgenic animals used in research. Detection of phytopathogens. The agricultural industry is constantly striving to produce plant propagules or seedlings that are free of pathogens in order to prevent economic losses and safeguard health. Systems have been developed that allow detection of small amounts of the DNA of Phytophthora, an oomycete that kills oaks and other species, mixed in with the DNA of the host plant. Discrimination between the DNA of the pathogen and the plant is based on the amplification of ITS sequences, spaces located in ribosomal RNA gene near unregistered trademark S coding area, which are characteristic for each taxon. Field-based versions of this technique have also been developed for identifying the same pathogen. Detection of genetically modified organisms. Qpcr using reverse transcription can be used to detect GMOs given its sensitivity and dynamic range in detecting DNA. Alternatives such as DNA or protein analysis are usually less sensitive. Specific primers are used that amplify not the transgene but the promoter, terminator or even intermediate sequences used during the process of engineering the vector. As the process of creating a transgenic plant normally leads to the insertion of more than one copy of the transgene, its quantity is also commonly assessed. This is often carried out by relative quantification using a control gene from the treated species that is only present as a single copy. Clinical quantification and genotyping. Viruses can be present in humans due to direct infection or co-infections. This makes diagnosis difficult using classical techniques and can result in an incorrect prognosis and treatment. The use of Qpcr allows both the quantification and genotyping of a virus such as the hepatitis B virus. The degree of infection, quantified as the copies of the viral genome per unit of the patent unregistered trademark S tissue, is relevant in many cases. For example, the probability that the type 1 herpes simplex virus reactivates is related to the number of infected neurons in the ganglia. This quantification is carried out either with reverse transcription or without it, as occurs if the virus becomes integrated in the human genome at any point in its cycle, such as happens in the case of HPV, where some of its variants are associated with the appearance of cervical cancer. References. Bibliography. Elise Poitras et al. Anhoud. La PCR en temps ra copyright L. Principes et applications. Reviews in Biology and Biotechnology 2:2-11. Boston S.A. Absolute quantification of mRNA using real-time reverse transcription polymerase chain reaction assays. J. Moll Endocrinal 25, 169-93. Higuchi, R. Dollinger, G., Walsh, P.S., Griffith, R. Simultaneous amplification and detection of specific DNA sequences. Biotechnology 10, 413 417. Holland, P.M., Abramson, R.D., Watson, R., Gelfand, D.H. Detection of specific polymerase chain reaction product by utilizing the 50A. 30 exonuclease activity of Thermus aquaticus DNA polymerase. Proc. Natal. Acad. Sci. USA 88, 7276 €7280. Qpcr M, Andrade J. M, Bengtson M, Foruten A, Junak J, Lind K, Sandelka R, Stjabak R, Stjogreen B, Strumbom L, Stahlberg A, Zorik N. The real-time polymerase chain reaction. Mol Aspects Med 27, 2 to 3, 95 to 125. Higuchi, R., Fokula, C., Dollinger, G., and Watson, are kinetic PCR, real-time monitoring of DNA amplification reactions. Biotechnology 11-10-26 €1030. Fillion, M. Quantitative Real-Time PCR in Applied Microbiology. Callister Academic Press. ISBN 978-1-908230-01-0, Warwick B., Paul J. H., Tabita F. R. Real-Time PCR Quantification of RBCL mRNA in diatoms and pelagophytes. APPL. Environ. Microbial. 68, 3771-3779. Logan J., Edwards K., Saunders N. Real-Time PCR, Current Technology and Applications. Callister Academic Press. ISBN A978-1-904455-39-4, External links, Beginner's Guide to Real-Time PCR by Premier Design, the reference in Qpcr Academic and Industrial Information Platform, Real-Time PCR tutorial by Dr. Margaret Hunt, University of South Carolina, September 5, 2006, OpenEtwer. Ref Genes Open Access Online Tool to identify tissue-specific reference genes for a Qpcr, Real-Time PCR User Experiences, articles about real-time PCR.